So now we get to our final stage, which is oxidative phosphorylation. Now the phrase oxidative phosphorylation refers to this idea of oxidating something, which means to remove electrons from it or to strip electrons from that substance. And then phosphorylate means to attach phosphate group to something. So oxidative phosphorylation is a comprehensive stage that starts with the oxidation of NADH and FADH2 and ends with the phosphorylation of ADP to make ATP. So remember, our end game is to make ATP, which just means throw that third phosphate group back on ADP. So we're going to see that here in this final stage where we get most of our phosphorylation done. You'll recall that very little ATP is made in glycolysis. Only 2 ATP are made per glucose, and only 2 ATP come out of the citric acid cycle. But here we're going to get about 30 ATP. So this is going to be our big ATP payout. Now, oxidative phosphorylation is not a typical metabolic pathway like you saw for glycolysis in the citric acid cycle. Instead, it relies on membrane proteins that are both doing some chemical reactions and doing transport. So let's go ahead and draw out oxidative phosphorylation. So we're going to start our review of oxidative phosphorylation by looking at the mitochondria, specifically the inner membrane of the mitochondria where oxidative phosphorylation occurs. So the inner membrane of the mitochondria breaks up the mitochondria into 2 compartments, the matrix and the intermembrane space. Now the inner membrane is going to have a series of proteins that are embedded in them. Some of those proteins are involved in transferring electrons from one protein to another, and therefore those are referred to as the electron transport chain. Now please note that ATP synthase is not part of the electron transport chain. I have 4 proteins drawn here. There's actually more than that, but we're going to simplify and assume there are 4 proteins in our electron transport chain. Oxidative phosphorylation is going to take the potential energy that we stored up in glycolysis and the citric acid cycle in the form of NADH and FADH2 and convert that potential energy into the phosphorylation of ADP to create ATP. So that's our goal, convert the potential energy of NADH and FADH2 into ATP energy. Now it starts with these electron shuttles or coenzymes, NADH and FADH2, coming up to the membrane protein in the inner membrane and depositing their electrons. So this process of depositing the electrons, which in our video was represented by softballs, into this protein is known as oxidation. So we're oxidizing NADH and dropping off the electrons into the protein. And then those electrons are getting passed from protein to protein. Now you might remember that the goal is not just to play past the electrons, that doesn't do anything, but that electrons are negatively charged. And so when they're negatively charged, they attract positively charged things, like H+. So the electrons are going to be responsible for pumping across hydrogens, ions, into the intermembrane space. And what that does is create a gradient of hydrogen ions in the intermembrane space. In fact, as you might expect, the accumulation of hydrogen ions makes the intermembrane space actually quite acidic because there's an excess of H+. So our goal is actually to create a hydrogen ion gradient in one of the compartments. And this is going to drive the production of ATP. While NADH deposits its electrons into the first protein in the electron transport chain, FADH2 deposits its electrons into the second protein. Now, because it starts later in the chain, FADH2's electrons do not pump as many hydrogen ions across as NADH. In fact, FADH2 has less potential energy than NADH and therefore contributes less to the hydrogen ion gradient. Now that we've looked at that, let's take a look at what happens when the electrons reach the end of that chain. Here is where oxygen comes in. So oxygen is known as the final electron acceptor. And what that means is oxygen is responsible for yanking those electrons off the chain and therefore allowing the continuous flow of electrons through the chain. So let's briefly take a look at the chemical reaction involved. We have an oxygen molecule, which is 2 oxygen atoms. And what's going to happen is they're going to actually pull off 4 electrons in total. And then it's going to combine with 4 hydrogen ions that we don't know where those are yet, so don't worry about that. There's 4 hydrogen ions in the solution, 4 electrons from the electron transport chain, and together these all combine to make 2 different water molecules. So oxygen pulls the electrons off and becomes water. Now, the reason why this is important is because in order for the hydrogen ion gradient to continually be propagated, electrons have to continually flow through the chain. They cannot be static and just sit there. So it's super important that oxygen pull the electrons off the chain so the electrons from the 3rd protein can move into the 4th protein, the ones from the 2nd protein can move to the 3rd protein, etc. The analogy that I want to give you is one like a highway. Imagine if you had all of these cars on the highway and you want to add more cars to the highway. Well, in order to get more cars on an already jam-packed highway, you better have some cars leaving via exit ramps. So, in other words, in order to promote the continual flow of traffic, some cars need to be leaving via exit ramps in order to allow more cars on the freeway. And the same is true here. We're going to have to pull the electrons off the chain in order to allow NADH and FADH2 to deposit new electrons to the chain and therefore continually pump hydrogen ions across. So now let's go ahead and add in ATP synthase into our inner membrane in the mitochondria. Now ATP synthase is a special membrane protein that has 2 personalities. It is part enzyme and part channel protein. It is a channel protein for hydrogen ions. So it allows hydrogen ions to diffuse down their concentration gradient. And since they are concentrated in the intermembrane space by the proton pumps of the electron transport chain, they want to diffuse down ATP synthase into the matrix. Now, along the way, they kind of tickle ATP synthase and cause it to contort in really weird ways. And what that does is that takes ADP, which is negatively charged, and the phosphate group, which is negatively charged, and brings them together in kind of a weird contortion. So I like to think about this like I'm pretty ticklish, and so if you tickle me, I'm going to contort pretty forcefully in really bad ways. And so that's basically what this enzyme does. It takes 2 negatively charged things that don't want to be together, and it forcefully contorts them so that they do come together, and then that links them together and creates ATP. So the hydrogen ions coming down the channel protein cause some pretty serious contortions in the enzyme that allow it to bring ADP and the phosphate group together and create that magical currency known as ATP. So it's really an ingenious design. So this summarizes our process of oxidative phosphorylation. Again, we're converting the energy that is stored in NADH and FADH2 into a hydrogen ion gradient that is then used to fuel the phosphorylation of ADP via ATP synthase. Now, the whole thing is driven by oxygen because oxygen is required to pull the electrons off the electron transport chain. So this begs the question, what happens if there's no oxygen? Then what happens? So of course this happens when our cells are starved for oxygen. And in order to answer this question, we need to understand that there isn't a single NADH or a single FADH2, but rather hundreds of them that are all waiting their turn to deposit the electrons in the electron transport chain. And if there is no oxygen, then the electron transport chain becomes jammed because the electrons are not being pulled off, and so therefore they are not jumping from one protein to the next, and therefore there's no spots at protein 1 or protein 2 to accept new electrons from NADH and FADH2. So as a consequence, the NADHs and FADH2s that are sitting there like cars waiting to get on a freeway just sit there. So one outcome is that all of the NADH and FADH2 are in their full or reduced form. So there's no available oxidized NAD+ or FAD. But in addition, the electron transport chain stops. And when the electron transport chain stops, it's not pumping hydrogen ions across. So the hydrogen ion gradient needs to be continually replenished in order to compel H+ to go down ATP synthase. So if you're not continually pumping hydrogen ions across into the intermembrane space, then you're going to lose your gradient because H+ will reach diffusion equilibrium, and therefore no more H+ will diffuse down, and you will stop making ATP. So without oxygen, ATP synthesis is lost in the oxidative phosphorylation stage. Okay, so we know that if oxygen is not present in the mitochondria while oxidative phosphorylation is being performed, that oxidative phosphorylation will just stop altogether because the electron transport chains become jammed and therefore unable to accept any new electrons. When they're unable to accept new electrons, the pumps stop working, and therefore the hydrogen ion gradient is lost. Without a hydrogen ion gradient, there's nothing compelling hydrogen ions to travel down ATP synthase, and therefore ATP synthase is not actually running as an enzyme because there's no hydrogen ions fueling it. So what can we do? Well, if oxygen is absent, we can still run glycolysis as long as we add in a modification at the end. And that modification is known as lactic acid fermentation. So glycolysis, as we've already learned, give us 2 ATP per glucose, which is not a whole lot. That's a lot like making $2 per hour. It's not going to sustain you for very long, but it is a stopgap measure that at least is better than nothing. Also, it can be produced really quickly. So as we're going to learn, some cells actually prefer to use glycolysis as their main ATP generating method simply because it's really fast. You get your money like that. It's like boom, 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 boom. Here's your ATP. You don't have to wait for it. But you burn through your glucose really fast when you do that. So what we're going to do is take a look at this modification that we have to add at the end of glycolysis. But wait, I didn't think that glycolysis used oxygen. And you're right, it does not use oxygen. But something else happens when we lose our oxygen from our cells. So here's that really complicated picture of glycolysis. And what I'm going to do is kind of go through this process again. You don't need to write any of this down. But what I want you to do is follow the steps. So remember, this is a metabolic pathway. And because it is a metabolic pathway, that means these steps have to happen in this order. So we start with glucose. We have our ATP investment phase. This is where we're actually spending ATP to make glucose unstable. So we go from glucose. ATP adds the phosphate group to make glucose 6-phosphate. Then we're going to change it into something else. Now we have to spend even more ATP, and finally this molecule is unstable enough that it's pretty easy to snap in half. And so that's exactly what you do. You snap it in half and you get 2 halves that are pretty much identical. And we learned this as glyceraldehyde 3-phosphate, or G3P. Okay, so now that we have this molecule right here, this glyceraldehyde 3-phosphate, now we're finally going to get our money back. So you can see down here, we make ATP and we make ATP. But before we do that, we have this really big step here, which is taking an NAD+ and grabbing a hydrogen ion and 2 electrons and becoming NADH. So this step has to happen before you can make your ATP. That's critical. Well, remember the problem from oxidative phosphorylation? If your electron transport chains are jammed, then you do not have available NAD+. You only have NADH available because it's sitting there waiting to be distributed into the electron transport chain. So what does that mean? That means that actually we can't even run all of glycolysis. We start with glucose and we can make it down to this part right here, glyceraldehyde 3-phosphate, but as soon as we try to do our next step, we are missing the important coenzyme that we need to be able to run this chemical reaction. In other words, the enzyme that does that step cannot do that step without its coenzyme NAD+. And we don't have any NAD+, because the NAD+ is all in NADH format, which again is like a full taxi cab, and we need an empty taxi cab. So what do? Well, we basically decide that we need to move forward with glycolysis no matter what. And so we're going to wastefully just throw some electrons and a hydrogen from NADH onto some other molecule that's pretty abundant, and so that we can free up some of our coenzymes and make them NAD+ again. So let's take a look at how that happens. Here's another look at glycolysis, and you can clearly see that NAD+ is required for G3P to move to the next stage. Now typically this is a good thing. This is a lot like taxi cabs picking up passengers. But there's a finite number of coenzymes, and there's a finite number of taxi cabs. So imagine if all of the taxi cabs in a local area were full of passengers, and there was nowhere to dump those passengers off. Well, you can't transport new passengers until you get rid of the old ones. And the same is true for the coenzymes. If all the coenzymes are in their reduced form, and they have nowhere to dump off their electrons, then they can't pick up any new electrons. So that's the impasse that we're at. We want NAD+, we want the empty taxi cab to be able to move through glycolysis. Well, typically, the taxi cabs are dumping their electrons off at the electron transport chain. But without oxygen, that electron transport chain is jammed. So what we're going to need to do is find another place for those electrons and that hydrogen on NADH so we can free it up and make NAD+ again. So that's our goal, is basically to dump NADH's electrons and hydrogen somewhere to make NAD+ without oxygen. So the solution is actually to dump them onto a pretty common molecule that is building up, and that is pyruvate. So remember, pyruvate would normally get shuttled into the mitochondria where it would be further broken down, but the citric acid cycle is jammed as well. So pyruvate is accumulating in the cell with nowhere to go, so we're going to make it useful by dumping the electrons from NADH onto pyruvate. Of course, this is without oxygen. So when we redraw the structure of pyruvate, we're going to see that NADH drops its electrons off onto the middle carbon, and in the process becomes oxidized, which is our whole point. Now, when we do that, you can see the middle carbon there is going to take a hydrogen from NADH. And so when that happens, carbon then has 5 covalent bonds, which is not possible. So what we're going to do is we're going to take that oxygen that's attached to that carbon and make it also take a hydrogen so that it can become an alcohol group. And so then what we have here is lactic acid. So the final product of the reduction of pyruvate by dumping the electrons and hydrogens from NADH onto pyruvate is lactic acid. So lactic acid gets built up in the cell when there's a lack of oxygen, because pyruvate is being converted into lactic acid by this process of oxidizing NADH. So remember that the goal, though, isn't to make lactic acid. Our goal is simply to shove our electrons and hydrogen somewhere so that we can have an empty NAD+ or an empty taxicab again. So again, lactic acid fermentation's goal is to oxidize NADH in the absence of oxygen so that glycolysis can continue, so that you can at least get 2 ATP per glucose even when there's no oxygen. All right, that concludes our lecture.